Hi, good afternoon, everyone. Welcome to Temasek Life Science Accelerators live webinar. I'm Hong Hui, your moderator for today. TLA is a joint venture between the Temasek Life Science Laboratory and Vertex Venture Holding. As one of the first deep bio-agritech incubator, TLA has set up to catalyze and nurture disruptive life science innovations into successful enterprises. We also run a pre-startup program, which we call the Hatchery, to focus on nurturing and matching novel ideas and entrepreneurs to support fast-growing and exciting innovation ecosystem here in Singapore. Today, we believe it will be the innovation that will eventually bring us out of the COVID-19 crisis. And this webinar is one of a series that we are planning. We hope to give innovators a platform to introduce their ideas to a wider audience and to see how we as a community can work together to bring about greater impact. COVID-19 has uncovered so much about ourselves as individuals and as society. The restrictions imposed on our everyday lives stem from the uncertainty of not knowing. Countries have responded quickly, do their best to find the right model. For example, in France, a French consortium just announced on Monday the launch of producing a saliva-based screening test so as to detect the SARS-CoV-2 virus with less than 1 milliliter of saliva from a person. But it is still unclear how much testing is needed. This sort of reminds me of a popular Minesweeper video game that we used to play when we were younger. It's detecting the COVID-19 requires clues about the number of cases in each field. The only way for us to ring-fence the COVID-19 virus is through proactive and widespread testing. It is our best window into how the pandemic is spreading. So over here, I wanted to share with you all a chart, a graph that just came out yesterday. This is actually a graph that's about the current testing status. Sorry, it jumps. It's the current testing status that we are seeing globally. It actually shows all the way from February until May for the last 3 months. And it measures the number of tests that each country is doing per 1,000 population. As you can see that the first wave of testing came when the first wave of the country was being hit. This is where the countries like Korea, and followed quickly by the second wave, like the US, Germany, and even Russia. And despite this is not, by the way, this is not measuring the total number of tests. It's a test per 1,000 people. You can see the intensity of the test actually increases significantly over the time. This showing that more and more emphasis the countries are putting on tests. So today, in the next 35 minutes of this panel, we will be looking at some of the key discussion topics. For example, we'll cover what is the general COVID-19 diagnostic landscape. We're going to discuss the limitation and challenges that's faced by the current state of COVID-19 testing. And what are the new innovations that is emerging in this space, and what are their challenges? And finally, how can we make this widespread testing readily available by bringing physics, engineering, biology, and machine learning together? We'll find out soon. And before we start, shall we just do a quick audience poll? Can you share with us, where are you currently dialing in from? Wow, most of us is from Asia, and we do have people dialing from Europe and Americas as well. So without further ado, please join me in welcoming Dr. Ian Chon, Chief Scientific Officer and Chairman at Pavnova Laboratory in Singapore, and Dr. Anthony Boulanger, Chief Executive Officer and Founder, Green Tropism in Paris. Hi, Anthony.
Hi, everyone.
So, Ian, can you tell us more about yourself and Pavnova?
Thanks, Jack. So yeah, depending on whether you're a lawyer or a scientist, you probably would think I'm a traitor or a reformed lawyer. So yeah, my past life in 2000, I was a lawyer at Harry Elias in Singapore, and we did corporate criminal law. It was very, very exciting. But I also had a lot of clients who were scientists, and I really, really got irresistibly drawn into their work, to the point that I felt I was on the wrong side of things. You know, I didn't want to represent them. I want to be them. So I ended up going back to school. I went to grad school at Johns Hopkins, the PhD program in cell and molecular medicine. I did my postdoc there as well. And then I spent some time in Uppsala University in Sweden, doing some algorithmic work on the invitation of a professor there. And in 2014, I was connected with Professor Chan So-ha. So we collaborated, and I was using AI to automate his IFA for NPC detection. Then in 2017, we incorporated PathNova and started offering a clinical service for NPC detection. And currently, we are extending our AI platform to autoimmune disease, and more recently, and very relevantly, to COVID detection as well.
Well, that's a long journey and so many twists in between. Very exciting. And now back to Anthony. Can you tell us more about yourself and the work that you're doing at Gene Tropism?
Yes, with pleasure. So yeah, indeed, it's pretty much like rainbows. It's a photonics-based technology. Before going deep into the explanation, yes, maybe a few words about myself and Green Tropism. So I have a PhD in microbiology and biostatistics that was applying to environmental processes. And then I was working for more than 3 years in the big French group about wastewater treatment plant and waste management, in order to be able to perform control monitoring of the process with near-infrared spectroscopy, actually. The goal was really to be able to provide to the company the means to perform non-destructive real-time analysis. And then when I saw what we can achieve with artificial intelligence when it comes to spectroscopy, I launched my own company, Green Tropism, and the core activities of the company in terms of research and business is to be able to bring to the users and to the manufacturers of spectrometers artificial intelligence algorithms in order to process the data in a fully automatic way. This way we can spare time of development, and especially we can detect so many things that are useful for the people and for the industrial guys.
Great, thank you. And okay, so Ian, currently, as we're seeing our current battle against the pandemic, it sort of reminds me of this Death Star in the Star Wars. We must find, analyze the blueprint and find a weak link in the SARS-CoV-2 virus. Where, in your view, are we now in this battle? What are the types of fighters, I mean, the diagnostic kits that we have actually to help us in this fight? And can you also break down some of the different type of tests that's available for us?
I think that's a very good question, Jack. So the question being, what's the motivation for testing? And how do we understand it? And what tools do we have to actually perform testing? This is a very common question. I think that I like to use the language that epidemiologists use. Most epidemiologists will be modeling the outbreak on something called the SIR model, or the SIRD model. And I like that language because SIR is an acronym, and S stands for susceptible, I stands for infectious, and infected, and R stands for recovered. So if you consider every living person in Singapore, you're either susceptible, infected, or recovered. And the mental experiment is, if we had perfect knowledge of everyone in Singapore or the world as to which category you're in, S, I, or R, this pandemic would be over, right? Because you would be able to treat each category appropriately, confine the disease, and then we wouldn't have a pandemic. So in a way, this is like a a dichotomy between perfect knowledge and imperfect knowledge. And right now we have imperfect knowledge. In a way, this pandemic has been framed as a medical war, right? A war on a virus. But it's actually very useful to think of it as an informational war, where information gives us the upper hand. Now, unfortunately, so in a perfect knowledge world, we'll be playing chess, like in maybe something closer to SARS-CoV, the first SARS-CoV. You have symptoms, that's when you're infectious. We have perfect knowledge, we can stop this outbreak from progressing. Now, the problem with SARS-CoV-2 is we're playing battleships. We have no idea where everything is. So we're just lobbing things randomly many times and hoping to get more information about, oh, is it the aircraft carrier here, or did we hit a destroyer, right? And then feel away from there. So testing gives us information, and Jack, to address basically the tools that we have largely two kinds of tools. And the first tool is to test for people who are currently infected. So that's the I part of things. So mostly for that purpose, we'll be using something called RT-PCR. And it's used in two ways, I guess. You could say that we use it to screen for new infections. And in that case, scale and speed are very important. Scale because we need to test a lot of people, and speed because it has to be actionable. And then we use RT-PCR in yet another way to verify that people who are currently infected are no longer infectious. So if someone has COVID, then you can watch their viral load decrease over time. And once they have, say, 3 consecutive negative tests, you clear them. Okay, so that's the first bunch of tools. Second bunch of tools is to figure out who is in the R category. So the people who are in the R category, usually we test for them mostly using serology like ELISAs and lateral flow assays. And we want to discover who had the disease, and we discover that by testing for the antibodies that they make to the virus. Now this also has a very important, answers another question, which is how many people were asymptomatic? Maybe someone didn't show any symptoms, but you find antibodies. That's good evidence that this person had seen the virus before, but just did not show symptoms. So for these kinds of tests, they're pretty important for return to work programs. And if you think about healthcare workers, especially if one of them had COVID and you confirm that they actually had antibody response and neutralizing antibodies, perhaps, then they can safely go back to work with confidence that they have some kind of immunity. So what's different here is with COVID-2, due to this phenomenon of pre-symptomatic, asymptomatic spread, our testing tools are kind of inadequate. And that's why social distancing is so much more important this time around compared to SARS-CoV. So I hope that covered what you were asking.
Yeah, clearly there looks like we actually have a lot of testing to be done, both for the infected, for the recovered, and maybe the combination of both. Sometimes you just don't know what state of the patient is. So with all these tests that we have to do, what are the limitations and challenges currently we are being faced with this current state of testing? And how is the world actually currently addressing these challenges? Ian and Anthony.
Yes, there are several ways to answer that, because as you already talked about, one of the limitations is what is the kind of test I want to put for my people according to the size of my population, according to the facilities I have, and according to the amounts of reagent, for example, I have at my disposal. If we take the example of RT-PCR, RT-PCR is a very powerful technique. When the RT-PCR says that you are positive to a certain strain, you are positive. But still, when the RT-PCR says that you are negative, you can still have some kind of doubt because maybe the sampling events that goes from the patient to the sample has not been done in a perfect way. You didn't go into the biological compartment where the virus was at the moment of the infection, for example. So that's one one difficulty for that. The other could be very practically the amount of swabs and the amount of reagents you can have. Because for RT-PCR, but it's true for other kind of tests, you need some material, so like swabs, like reagents that are not available at all time in all place, because the urgent need that we have for the SARS-CoV-2 detection was so huge across the world that you have some place in the world that were lacking of this basic material in order to be able to test the population, or even to test some people presenting symptoms that could be from SARS-CoV-2, that could be for other viruses, and it's not the same way to treat these people.
Limitations and challenges, just keep it simple. I think there are two elephants in the room that are not really covered often enough by the press. I think that one of them actually is an echo of what Anthony just said. So the first elephant is that there's this false negative rate of RT-PCR. No one talks about it enough, right? So the false negative rate for the first 5 days after exposure of someone to the virus can be as high as 67%. So that's 2/3 of people, 67. So that's like, this is a very recent recent meta study that looked at least 1,000 patients. And so you can't trust testing before day 5. The lowest false negative rate occurs somewhere around day 8 after exposure. So that's about, and that false negative rate is 21%. It's 1 in 5. So it's still not at 0. So it's pretty high. This is something which is, I guess, RT-PCR's deep dark secret. And I actually knew someone who actually was a visiting professor in Wuhan during the, he was a clinician, and he was in the middle of outbreak in February going to March. And they had given up on the RT-PCR because they were just going on the CT scans, because RT-PCR at that point had a 70% false negative rate, when the patient clearly had all the signs of COVID. Yeah. So I think this is one serious problem. So, while I think our current thing for screening has been to focus on making RT-PCR better. So we have POC versions of RT-PCR. We are working on optimizing the transport medium and also the MP swabs. All these are very, very good and important innovations. But if we are talking about reasoning from first principles and talking about what will lead to a real breakthrough, then it must come from solving the false negative rate for RT-PCR. The second elephant is actually kind of the first elephant version for serology. So the first elephant was the false negative rate for RT-PCR. The second elephant is the false positive rate for serology. And here there's a very, very interesting story, which is Stanford University very recently did this study in Santa Clara County. And they found that they tested a large number of people, and they found that people who had never shown symptoms of COVID, 4.16% of them were serology positive. So they concluded that all of these individuals were essentially asymptomatic. And that when you extrapolate to the US, a lot of people walking around who actually had the disease before, but were just asymptomatic. Now that turned out not to be true, because if you had taken the false positive rate into account, these patients were very, very likely mostly just false positives. So when talking about serology, you need very near perfect specificity for serology to be useful. And this kind of mirrors the conversation we had just before we went live. And our current thinking for serology is focused on, again, making ELISA lateral flow better. We're making different versions of it. You can even do it at home. Now, all these are very good. They're very important. But again, if we're talking about from first principles, I think a real breakthrough must come from solving the false positive rate for serology.
Yeah, so that's kind of like, is it true because of this false positive and false negative rate? That's why sometimes a patient had to go through multiple tests before they can be confirmed?
Absolutely.
Yeah.
Absolutely. In actual practice, one test is not enough. And so because our information is imperfect, so we need confirmation. And that's why even to clear a patient, you need to have 3 consecutive negative tests.
I see.
So we solve for these 2 elephants. I think then the strategy can, then becomes making these solutions as fast, portable, and robust as possible.
I see. So I remember, during the last, I think all these people, especially in Asia, vividly remember during the last 2003 SARS outbreak, this thermal scanner was being introduced. And until now, this has been the main way of testing people at the various places, the public places, that you're entering buildings and all that. So do you think that post-COVID-19, we will actually to see a new, maybe a hyperspectral scanner that can be used to tell at an instant whether someone is actually carrying this SARS-CoV-2 virus, even if they are asymptomatic? Anthony? Perhaps Raman?
Yeah, maybe perhaps Raman. It's what we are looking for. Maybe a few words about Raman, how it works, and what is the main principle and how to work with that. So maybe the audience has already heard about spectroscopy, spectrometry. There are a lot of various techniques, and the basic principle is pretty much always the same. When you want to analyze a spectrum, you will shine a light on the spectrum, and according to the level of interaction between the light and the matter you are analyzing, you will have what is called a spectrum. That is basically peaks among the graphic. And in fact, this spectrum is very important in terms of data and in terms of information, because it is carrying all the information about the composition of the sample. So we are looking, for example, at mass spectroscopy. You will use the spectra and the information about mass and time of flight in order to be able to tell, okay, this is this molecule, this is this cocktail of molecules, and so on. If you are looking at some vibrational spectroscopy like near infrared, for example, that is widely used in industries, it will rely on the level of interaction between molecules, the links between atoms, and the light. And for Raman spectroscopy, it's a bit different. It's still spectroscopy techniques. What you will do is to shine the sample with a laser, monochromatic light, and you will have a look on what is called the Raman shift. So let's say the shift from the light because of the interaction between the electron clouds of the sample, because of the composition of the matter that you are looking at. And since every sample is different in terms of qualities, in terms of molecules, in terms of quantities of these molecules, you can say, okay, this Raman spectrum is defined for these molecules, or for this amount of molecules, or this mix of molecules. And at the end of the day, what you will have, it's a database of spectrum. But still, you need one step in order to go from a spectrum to a valuable information, that is a machine learning or artificial intelligence, in order to be able to make some correlation between what you have in your database and what you want to see at the end of the day. So for example, for this SARS-CoV-2 topic, the philosophy is the same. You have to build a database from patients that are infected by the virus, patients that are safe from this virus, and then to apply artificial intelligence on this database in order to see, okay, when I have these patterns of spectrum, when I have this correlation between this wave number and this wave number, I can tell, okay, you are positive or you are negative. And artificial intelligence is really a key in this technology. This is a topic we are working for more than 5 years now, not only on the Raman spectroscopy, but on other kind of topics, because it's not so easy mathematics. You will deal with some artificial neural network, for example. You will deal with some nonlinear combination between the variables, but you need this amount of mathematics in order to, as Jan said, to be sure of your answer. You don't want to miss the SARS-CoV-2 virus, and you don't want to have a mismatch between, let's say, SARS-CoV-2, SARS-CoV-1, or other respiratory viruses. So to come back to your first question, I do believe that the ultimate goal is to have this scanner that is able to rely on artificial intelligence and photonics as Raman in order to provide pretty much instantaneous analysis with a non-intrusive way to take the samples from the people.
I see. Maybe Ian, do you have something to further to add on? Because just now we were talking about some of the constraints of the existing tests, especially in terms of the speed, the precision, and also the portability. But I remember PCR, have to be brought back to the lab for testing. So what do you think that Raman can solve these issues?
Right. So first to add on to what Anthony said, and then to address those questions. So Anthony mentioned that Raman, basically it looks at the entire composition of the sample. And I think that's a game changer. Of course, with Raman is actually a data-driven platform. You're not testing for one thing, you're looking for a signature of all the chemical components that are actually in the sample itself. So you're not just testing for the virus, you are also getting information about the patient's response to the virus. I believe that information will implicitly be very useful in coming to a diagnostic decision. So by considering more information in the virus, you can in principle solve the false negative rate problem for RT-PCR. And then to the points that you're alluding to, a Raman device is not an RT-PCR device that has to cycle up and down many times and takes like even the fast versions take like an hour. So you're talking about a device that collects spectra on the scale of minutes, could be a minute. It's non-destructive to the sample, so you can still do RT-PCR after you have taken a Raman spectrum. It's very portable. You can fit in your hand. There are also portable versions that fit that you can carry around and fit on a desk. They're a bit like a little radio. So you can set them up anywhere. You can deploy them anywhere that you have a flare-up, hotspots, nursing homes, ports of entry. I think that ability to deploy at a very granular scale and be very responsive on site, and at least get a first screen of all the people who might be positive in a certain area, is a game changer that Raman can achieve.
So does the patient still require to be, first of all, getting the specimen through the nasal swab? Because that has been a really invasive way of getting it. Some people find it very uncomfortable. Can it be true, like maybe a saliva or something to collect the specimen?
I think Anthony has better information on this.
Yeah, Anthony.
Yeah, yes, indeed. We are currently going through tests in order to compare the nasopharyngeal swabs and the saliva swabs. It's really promising at this step of the work because what is really interesting with Raman, it appears to be more sensitive than the RT-PCR. So even if you have a lower amount in your saliva, that the RT-PCR will miss because you don't have enough copies of the virus in this biological compartment at the very beginning of the manipulation, you can still have the chance to see that with Raman PCR. And when the concentration are high in your body, in your pharynx or in your saliva, it's for sure, it's way less intrusive to work with the saliva samples. This way it would be easier to test more people and to not rely on some intrusive test in order to get the sample and then to say, okay, I would use spectroscopy in order to tell you that you are non-infected indeed.
I see. Very, very interesting. So I'm just wondering, with yourself, and then yourself based in Paris, and then Ian based in Singapore, how did actually this collaboration between the two came about, especially within such a short time?
So basically, well, we were connected by Mr. Teo Min Kian, who knows both of us. He thought that we should explore working together. Now, I've been following the Raman technology space for a long time, actually, many years now. And I've also started helping some of the plant research groups in TLA who are currently applying Raman for automated monitoring of crops. And so when this collaboration opportunity arose, I was naturally interested, and especially Green Tropism's approach and Anthony's focus on software and being hardware agnostic, I thought was very innovative. I thought it was the right focus. There are a lot of companies that are trying to make Raman instruments, but it's just another Raman instrument. I think that the actual bottleneck is actually good software. So I think that, just to summarize, I think we are approaching our collaboration from complementary directions. Anthony has jumped straight into the clinical samples, and he's working top down, analyzing clinical COVID samples. And we are working from the bottom up, analyzing live virus using Raman, culturing live virus in BSL-3 to obtain a high quality Raman signature for SARS-CoV-2. And we hope this work will merge together and give a high quality Raman diagnostic.
I see. Anthony, anything to add on to that?
Yeah, I said already a lot and a lot of answer, but I fully agree. Obviously, what is really important in this term, in this kind of collaboration, because of the geographic constraints, because of the non-travel laws, and because of the time and the emergency, is I think that we match on the way to do science as well. Both of teams are fully enthusiastic and motivated to deal with the SARS-CoV-2 detection, and then other pathogens. We have to keep in mind that that things for additional collaboration in the future. And the way that we work, so in Paris we have indeed this clinical samples that are there. We can tackle several changes about the way to prepare the samples when it comes from real patients. What kind of hardware we should use, because there are a lot of things on the market, as Jan said, but software has to be the ones to manage the hardware and the samples. And the way to work in Singapore is really interesting, because it will feed the software as well, with pure signature of the viruses, with control condition for the manipulation, and that's why it's working today, because of this complementary approach of the two teams.
Yeah, once this the travel ban is over, and we're very much welcome you to come to Singapore. So eventually you can meet in person.
It would be a great pleasure.
Okay, so for now, let me just turn back to the audience a little bit. And everyone, can maybe I just ask you all a very straightforward question. I mean, after seeing all the discussion that we had just now, do you think that will COVID-19 screening become the new norm for traveler going forward? Means everywhere you go, you have to do this, even when this pandemic is over. Wow, most people, predominantly people, think that it will be a norm. And then with this, I'm sure that because currently for you to travel and to be tested, you need to be invaded with that nasal swab. Hopefully with this new test method that Raman is coming up for this new Raman method, and we don't have to do that anymore. So let me just have another question to all of you. This has to do with the challenges that we have facing now globally. So what do you think are the current challenges to a universal COVID-19 diagnostic platform? You can choose more than just one answer. There are total 8 different choices. Okay, so from the poll, it seems that the top 2 choices that you all see as a challenge is one is speed, one is precision. Yeah, I think these were the 2 things which we addressed early on. Ian, Anthony, what do you all see the feedback is? Yeah.
Yeah, I mean, I think I have to agree. I think that precision lowers the error rate and gives you information that's actually actionable without having false readings. And speed actually adds to the actionability, because we know that someone can be continuing to pass this COVID on without showing symptoms. So you want to actually have a quick result to be able to classify this individual correctly.
Okay. All right. So maybe another question to both of you. How do you think that the challenge of regulatory approval will be for this novel diagnostics that you're coming up? Or maybe you have any of the past experience that you can share with us?
It's something that we are currently have a look in France, as you can guess. And in fact, what happens, it's the way that this new technology is working is by using some photonics, meaning some physical phenomenon for analyzing the sample, and then using artificial intelligence tools in order to be able to decipher between positive, negative patients, or maybe uncertainty of on the results. So that's a new way for the guys we are talking with to have a result on diseases, because RT-PCR or serology, for example, are relying on actual biology, microbiology, and let's say that for the RT-PCR, when you want to see if someone is positive, it's because you have physical copies of the virus in the sample at the end of the reaction. Dealing with a photonic phenomenon is a different philosophy, and it's not difficult to explain or to make it acceptable for the regulations committees, but it's a new way to think, and that's really interesting. But it could be the way of the future to deal with new diseases. And I was really interested in the results about mutating viruses in the poll just before, because next year or in the coming years, we could have some mutation of viruses, and we will have to deal with that as well.
Sure. Ian, you have anything to add on to that?
Yeah, sure. I think our regulatory framework is relatively new. So I think the HSA has come up with actual guidelines for software medical devices, and that came out recently, December 2019, the most recent edition. And they have a section on AI medical devices. So that's good. At least there is a clear process to actually follow through. But I do not, this framework is relatively new to me. Probably everyone's feeling their way around. So I do not have good information on how quickly we will move through this regulatory framework in Singapore. Strangely, I have had more experience with the European side. So with Oncodia, the company that I was involved in, we went through the CE-IVD process. It took, was very smooth, very quick, 3 to 4 months we were done. There was also an AI software for calling cancer mutations. And I have to say, I think it's a very good process there that they have in Europe. And I'm hoping that Anthony will make our lives easier with it when he pushes it through regulatory in France. And that will also help us in Singapore.
That's great. So before we move to the Q&A, I'll probably reserve another 10 minutes for that. Just one last question. I think probably some of our attendees will ask that as well. So how far are we on the clinical trials for this new Raman spectroscopy method that we're using for the test?
Okay, I guess this one is for me.
Sure.
Yeah, clinical trials is ongoing and looks very promising. So in order to give more insights about that, we are testing various samples, nasopharyngeal swabs and saliva samples, because as we talked about, saliva would be the main lock to unlock in order to go on a fully non-intrusive way to detect viruses and so on. So we have actually very promising results on the nasopharyngeal swabs, and we are currently building the database for the saliva. What will be very interesting in the incoming days is to test additional viruses, especially respiratory viruses, in order to tell if we are fully specific of the SARS-CoV-2 strain or not. We have done this work for the nasopharyngeal swab, and we have seen that indeed each of the 15 viruses that we have tested has its own spectral signature. So it's very promising. What is really interesting is that even for negative patients that can have some viral strain on very low amount in the sampling, we can remove that as a, let's say, background noise with artificial intelligence. So we know that the combination of Raman with artificial intelligence is working for virus detection. And the next change is how to automatize that and how to robotize the whole process to go from the sampling with nasopharyngeal or saliva swab to the spectrum and to inject that into the artificial intelligence software to have the full process in 3 minutes.
Yeah, we can't wait to see that happen. Finally, to see that actually the test being rolled out. So now, without more waiting, maybe I will just turn to some of the question that's raised by the audience in the Q&A. For those that haven't done so, please do click on the Q&A button at the bottom of the screen to raise your questions. You can also upvote some of the other questions. So let me just take a quick look and see what we have. Okay, so we have quite a number here. And we actually have the one that's floating on top is, I think, from Amit. The question says is, why can't we extract antibodies out of the recovered patients and inject it in sick people? Is this treatment too expensive?
Okay, so I think you're referring to convalescent plasma, and that's actually something that's being done in Singapore too, I believe. And basically, I think it's a good interim solution. It's not totally scalable. I mean, at the end of the day, you also need to consider things about matching, for example, right? So, so I think it's not necessarily a very the best scalable solution, but it's a good option that we have right now for, especially for patients that are not doing too well. I think, yeah. So perhaps Anthony has got something to add here.
I fully agree with you. It's the philosophy of vaccine, maybe in a way related to the questions. It's not, as you can guess, my core skills, but I have the same feeling than you on this question.
Okay, so let's look at another question then. So are we actually looking at any specific target molecules via this Raman spectroscopy for detecting COVID-19? I remember when if we were looking at virus detection, there's different so-called features in the molecule that we're looking at. It could be the spike protein, it could be the N protein. So I'm just wondering what specific thing, I suppose this question is asking, the specific target that you're looking at for this Raman.
Okay, maybe I will start for this one. So indeed, we have to look at specific molecules because we want that our artificial intelligence model to be specific from SARS-CoV-2 detection and to be able to decipher between various strains. In the samples that we have analyzed, we have tested two things. We have tested the RNA coming just before the RT-PCR steps, and we have a signature that is linked to the RNA. And then we have gone to analyze the whole viruses after breaking down the constituents by the lysis buffer. And actually, we have additional signal in the spectrum that appears that are linked to proteins, that are linked to phospholipid, and still linked to the RNA of the viruses. So Raman spectroscopy is not targeting a specific molecule; is looking at the whole molecules that are present in the samples that you are analyzing. So this way, you can have a specificity coming from the proteins combining with the specificity coming from the RNA and the additional molecules that you can add in the envelope of the viruses, for example.
I think Anthony basically described it, and this is we're not looking at one thing like an ELISA would or RT-PCR would. You could say that this is actually doing all of that and possibly more. And Anthony's experiments in a way are revealing the layers of information that are actually being included in the signal. So this, I think this goes to the very data-driven nature of Raman.
Yeah, that would be great. So it actually looks like, we can actually scan through the whole sample and pick up any signal. It's not just relying on one or two features in the molecule, I suppose.
Yes, absolutely.
Oh, then that's very thorough in the scan. Or maybe now we can go to the next question. This comes from Sean Tan. Any comments on this isothermal nucleic acid diagnostic system, which can shorten the whole detection process? For example, the NEAR or LAMP isothermal technology?
Yeah, absolutely. Maybe just a few comments and Anthony can join after that. So I think LAMP is really great, and I think that it has a lot of potential, especially because of speed. I think that the use case for LAMP will be limited by the fact that it tends to be yes or no. So it's a binary result. So you either have a positive or negative. And right now, clinicians still use that quantitative result from RT-PCR for a lot of diagnostic decision making. Like they want to see the viral load decrease over time, and then before clearing the patient, right? Or they want to know how high that load is at the very, very start, so that they can try to figure out where in the life cycle of the infection the patient actually is. So I think LAMP can be very useful in certain contexts, maybe for quick screening, but it doesn't totally replace RT-PCR.
Actually, just now, I think Ian, when you say that, even I got a shock. You mentioned that the current RT-PCR test that a lot of people, a lot of the countries are using, it has a false negative rate of as high as 70%. Is that really that alarming? And also, how about its false positive rate?
That's a good question. I think Anthony did allude to the fact that if you have a positive result on the RT-PCR, you can probably trust it. But a negative result, a lot of false negatives there. Yeah, I think it is that alarming, and I think it's time dependent. If you look at every patient on a graph on a timeline, then the earlier you are in the infection, the more likely you are to have a false negative. But that's actually when you have the most effect on a patient's clinical outcome, by acting early, right? And also by confining the individual so that the individual doesn't spread anything COVID to another individual. So I think that the fact that you have these false negatives really highlights that the information that we're getting, we're in an information war, information we're getting is not great. There is uncertainty. So you can't trust it. So you keep pressing the button many times. You test multiple times just to be sure. And I think that pretty much sums up and highlights this problem that we're having with imperfect information for COVID.
Again, thank you. Thanks everybody for joining us at TLA's live webinar, and especially thank you to Ian and Anthony. And we are glad to actually see that the better diagnostics can actually help to light the path for us to go back to normalcy. I think most of us cannot wait to travel again. That's been holding us back to our seat for how many months. And so before everybody leaves the meeting, you will actually be directed to a post-webinar feedback form. It takes only about 2 minutes. I really appreciate that you all could give us some of the comments so you can help us to improve ourselves going forward in the future. And yeah, I think if you have anything, please email us. And for now, I think everyone stay safe and have a nice week.
Thank you, Jack. Thanks, Anthony.
Bye.
Thanks a lot.
Thanks, everyone.
Take care.